Hi guys, Andrea Spar here. I'm doing my presentation on the leucine zipper for Chem 330. What is a leucine zipper? A leucine zipper, also called a zip, is formed by amphipathic interactions between two alpha helices, one from each monomer. Amphipathic interactions are those caused by proteins having hydrophobic and hydrophilic components. The helices are bound together by hydrophobic interactions between leucine residues, which are located on one side of the helix. Due to tight packing of these hydrophobic side chains, the leucine zipper is a very stable structure. The photo above is from the book, and it also shows the CO group of each amino acid that forms a hydrogen bond with the NH group of amino acid that is situated 4 residues ahead in the sequence. What is a leucine zipper continued? A leucine zipper is created by dimerization of two specific alpha helix monomers bound to DNA. Basic amino acid residues such as lysines and arginines interact with the major groove of DNA, making sequence-specific interactions. And then to the side, you'll see a photo showing the major groove, the minor groove, and where the leucine zipper actually is. The photo is also showing DNA binding domain. The helix loop helix dimer is shown bound to DNA fragments. Each alpha helix represents a monomer. Did you know? The leucine zipper is a part of transcription, which is where DNA goes to RNA. It acts as a hydrophobic core for polypeptide chains that leads to the formation of protein. It's composed of tightly bound alpha helixes. An alpha helix is a coid coil that makes the leucine zipper very stable. This is due to the bonding of the hydrogen bonds, the ionic bonds, and other bonds. And this is found in alpha keratin, and examples of those would be wool and hair. Alpha helix. Alpha helices are polypeptides stabilized by intra-chain hydrogen bonds between amino and carbonyl groups, creating a coiled backbone. There are 3.6 amino acids per turn of the alpha helix. The alpha helix can frequently be seen in proteins. The leucine zipper domains consist of two motifs. A motif is a basic region that recognizes a specific DNA sequence and a series of leucines spaced 7 residuals apart of the alpha helix, typically in a repetic alpha helix turn helix conformation. The screw-like rotation describes whether the helix is right or left-handed, and ones that are commonly found in proteins are almost always right-handed because this helps the reduction of the steric clashes. Replacement. When trying to replace leucine residues, you can do so with isoleucine. Isoleucine contains a very similar structure to leucine. If you look below, I've posted a picture. Both contain an equal number of C atoms on their side chains. Therefore, both are likely to exert the same extent of hydrophobic interactions. Thus, replacement with isoleucine is most likely to function as efficiently as leucine for maintaining the hydrophobic core. And as for a destabilizer, replacing leucine with a charged amino acid is most likely to destabilize the hydrophobic core. When you compare the structures to the left, the isoleucine and arginine, arginine has a positively charged side chain and a total of 3 N atoms in the side chain that, being polar, can form hydrogen bond with any polar residue in the vicinity. Replacing leucine with arginine is most likely to destabilize the hydrophobic core because of the polar interaction at the core itself. Amino acid pair. Another set of amino acid pair that would be comparable would be aspartate and histidine. These would work with the electrostatic reactions. You would basically need one basic side chain and one acidic side chain. Your basic side chain is histidine, and your acidic side chain is your aspartate, and there's a picture of those to the right. And these will interact in the same manner, therefore producing the same structure, allowing the leucine zipper to still work properly. These are my references. Thank you guys for watching my presentation. I really appreciate it, and I look forward to the next 8 weeks in this class and reading everybody else's presentations. Thank you.